Hello, I'm Wei Ming, Associate Professor of Chemistry at Columbia University. It's fair to say that advances in biology and medicine in the past half a century has been greatly facilitated by the new emergence of physical tools, such as X-ray crystallography and DNA sequencing techniques. We now have complete genome sequence of many species, including as human, and the high-resolution crystal structure of macromolecular machineries, such as ribosome and membrane proteins. But to go from this kind of a static information to a full understanding of a living human being obviously requires much more in between. In particular, we need to understand those crucial biochemical interactions happening in space and time. Now, from many studies accumulated in the past half a century, we now appreciate that those crucial biochemical interactions are highly dynamic in time, heterogeneous in space, and complex in nature. So that is where imaging comes into the picture, help to resolve those crucial biochemical interactions in space and time, and help to make biology a quantitative science. So my group is working on developing novel imaging techniques and to using them to address biological questions. So one such area is called chemical imaging. As you can imagine that cells contain a large number of chemicals, including fatty acids, amino acids, nucleic acids, ADP, glucose, and neurotransmitters. So those small molecules, even though they are very tiny, they play indispensable roles in cells, particularly metabolism. However, they're extremely difficult to visualize. For number one, they are non-fluorescent to begin with. Number two, because they are so tiny, the conventional tools of labeling them by fluorophore actually does not apply here, because the large fluorophore will easily destroy the function of small molecules. So to address this challenge, my group is developing a unique advanced microscopy technique called Raman scattering microscopy. So basically, Raman microscopy combines the advantage of chemical specificity given by Raman scattering and signal amplification given by steaming emission. So the resulting Raman scattering microscopy will have both chemical selectivity and high signal detection sensitivity. So as you can see that my group is working on the interface of physics, chemistry, and biology. So we constantly harness the latest laser spectroscopy developed in physics community, and then in the lab we have synthetic tools to make new molecules, especially new imaging probes, and then we combine these two efforts together to address important biological questions, including lipid metabolism, brain metabolism, and cancer metabolism. Thank you.